for those of you who do not know me or have not had the honor yet of meeting, my name is Professor Jordan Wilcox, and I am a teaching faculty here at Notre Dame over in the Department of Psychology. And we here today are honored to welcome the inaugural Neuroscience and Behavior Distinguished Lecturer, Dr. Fiona Harrison. Dr. Fiona Harrison, yes, please clap. Dr. Fiona Harrison earned both of her bachelor's and doctorate degrees in psychology and behavioral neurosciences from Cardiff University in Wales, so she hails from the UK. You will get to hear her lovely accent. She is a principal investigator in the Division of Diabetes, Endocrinology and Metabolism in the Department of Medicine at Vanderbilt University Medical Center in Nashville, Tennessee. I have the particular honor today of introducing her as she served as both my graduate school and postdoctoral mentor, so we have a very long-standing and close relationship. She is an absolutely superb mentor to postdoctoral fellows, graduate students, undergrad students, high school students alike. And in addition to her own prestigious research program, she's also serves as the director of the Mouse Neurobehavioral Core at Vanderbilt University. She is the co-director of the Vanderbilt University Training Program in Environmental Toxicology. She is an active member of the Vanderbilt Brain Institute DEI Committee, an active collaborator with the Vanderbilt Memory and Alzheimer's Center and the Vanderbilt Kennedy Center, which is focused on intellectual and developmental disabilities research. So if you can't tell, she's fairly amazing, right? She has a long-standing list of achievements. Her research focus broadly is on diet and environmental exposures and how these influence our brain health, and particularly our susceptibility to developmental disorders as well as age-related neurodegenerative disorders. She's well known worldwide, in fact, for her work on the varied roles of vitamin C, I have learned it is pronounced, in the brain, and particularly the effects of that vitamin C deficiency. More recently, though, her work has taken a turn, and she's investigating other nutrients like manganese, which you will be hearing about today. So I can think that many of you will find her work particularly interesting and relevant to many of the things you're learning about in your own courses. And so we are absolutely thrilled to have her here with us. So please welcome me in joining her once again, Dr. Fiona Harrison.
Well, thank you, Jordan. That was very nice. I was slightly concerned about what she was going to say because she did tell a story at lunch to several students about a horrible, horrible thing I did to her during graduate school, which was to make her learn essentially how to set up an entire EEG system. So she's forgiven me now, though, and gave me a kind introduction. All right, so I'm thrilled to be here. I've never been to Notre Dame before. I've heard about it since I was in graduate school because I had an alum, and Notre Dame alum was one of my graduate colleagues. So I'm really excited to be here. And today's talk, I'm going to try and bring together several areas, including developmental disorders, environmental exposures, and manganese in particular. So I'm going to give background with some general concepts that I hope will be of interest to everyone, give you all something to keep you awake. And so I know that a lot of you are doing behavior and neuroscience, but that really you're more people heavy. And, human research is important too, but we focus on mice in my lab. So we're going to have a lot of mouse behavior in this talk. So I'm happy to be interrupted when people have questions. I don't promise to not go off on a tangent if you interrupt me, but please, if you want to ask questions throughout the talk, you are welcome to. Okay, so given that we are a lot of neuroscientists and psychologists in the room, does anyone know what these are in the picture? No guesses? Someone usually guesses neurons, and that some people get clever and they tell me that they're cerebellar neurons, something like that. But they're in fact manganese dendrites. So Jordan knows this because there are some of these in her office. So these are manganese deposits in the rock from Germany. And I just think this is a beautiful picture because they've always reminded me of neurons, and it just shows how important manganese is in the brain. All right, so everyone knows the phrase nature versus nurture, and we all know about gene-environment interactions. But what we're really talking about when we use those is the exposome or exposome, however you want to pronounce that. And essentially what that is how everything in this image here, and probably some other things beside, everything in your world comes together to influence how much you will be impacted by a disease or a condition in combination with your genetic risk. So this concept was first described, or the term was coined about 20 years ago by a researcher named Christopher Wilde. And it turns out that I've been studying the exposome for nearly 20 years without knowing that it was a whole realm of science. I just learned this term a few years ago. And I think it's really important to consider how many things in our world actually influence our brain health. So I've highlighted the ones that we're going to focus on in my lab, the ones we're going to talk about today. But there's actually a lot of things on here that we typically don't even think about. So things like walkability and green space where you live has a huge impact on health. And social and cultural norms, like at what point is it okay to go see a doctor or to ask for help? So there's a lot of very varied ideas that play into the exposome. But we're going to talk about something a bit more obvious today. Contamination of drinking water, air pollution is a similar idea. And usually in my lab, we're talking about diet. And in fact, manganese combines all of these factors together. So just in case this is new to anyone and to make sure we're all starting on the same page, what is the exposome? So we've all seen, this genotype leads to phenotype, but it's just a lot more complicated than that. It's basically any environmental or non-genetic drivers of health and then disease, and that's throughout your entire lifetime. So your genome and the exposome together are going to lead to phenotype. So there are some developmental disorders, and in fact, aging-related disorders as well, where a single genotype or mutation will determine whether or not you get that disorder. But in most cases, that's not the case. Most diseases or phenotypes are a lot more complex and have a polygenic background. And it's a combination of that polygenic risk as well as your environment that are going to determine whether you show the signs or the phenotype of that disease. And importantly, some of those factors, but not all of them, are under your control. So an easy way to demonstrate this is the concept of height. So there are several genes responsible for height. And in general, if you have two tall parents, you have a tall child. And if you have two shorter parents, you have shorter children. But there are other factors as well. So think about the child of two very tall parents, but the mother smoked or was exposed to some other toxins during pregnancy. That child might be smaller than its maximum potential height. The other example there, we have a child that may be developed under some kind of pressure like pollution or something, but still carries the genes for height. So when they have optimal nutrition during pregnancy, that child will come out taller than their parents. So there's a lot of different ways that we can affect the genetics and how genetics express. But this goes on throughout life. So height is actually really interesting because there comes a time when, after puberty, what you eat doesn't really impact your height anymore, just your width and breadth. But it doesn't really affect it. But the height that you reach can impact your entire life because a lot of psych studies will show us that people who are taller might be a little more, commanding of attention in a room, might be more likely to get promoted or get higher earnings and therefore have better health outcomes. So there's a lot of ways that developmental exposures can impact you throughout your entire life. Okay, and gene-environment interactions are particularly important for developmental disorders. The reason is that the brains in babies, fetuses, and small children are unformed. They're still dividing, they're still developing. This high rate of neurogenesis means that the impact of any mutations, any damage or epigenetic changes is going to be so much greater than in an adult brain. There's also an incomplete blood-brain barrier at birth, so there's more potential for toxins to impact brain function. Also, kids are stupid. They eat anything. They will put things in their mouth that they shouldn't. So they get exposed to a lot more than we do as adults. Right? We know that disorders like autism and ADHD have a highly heritable component. But even within families, there's a lot of divergence in how different disorders present. So here's one study from Sweden. And what they did was they looked at the basically the risk of autism that could be associated to genes alone, or to any combination of genes, or to other environmental factors, and found that about 40%, they were talking about how 60% was genetic risk, and that was their important point. But I'm more interested in the 40% that is not straight genetic risk. Okay, so that's other things in the environment or the shared environment that impact development of autism, for example. Now, ADHD is a little bit different. There are a lot of different subtypes of HD. There's really a lot of difference in severity levels. And there aren't really any single genes for this. So for the most part, we identify genes that do increase your likelihood of developing ADHD. So genetic risk is really a likelihood. It's not necessarily a definitive factor. And there's a lot of evidence that chemical exposures, prenatal exposures, parenting inputs, educational opportunities, there's a lot of different ways the environment can impact the expression of ADHD. And in fact, even just whether a child is diagnosed with ADHD, so socioeconomic factors, gender of children are going to impact, whether their behaviors are viewed as something that needs to go get treated. So really, the important take-home message here is that many common developmental and behavioral disorders are due to both genetic predisposition and environmental factors. The question is, given the really strong sensory angle to how a lot of children with autism or adults with autism spectrum disorder present, there's a lot of sensory changes, how can that be dependent on environment? So it's a great question, and I really hope to answer it throughout the rest of the talk, to give just one mechanism through which that could be. But the real answer to that is everything that you sense is based on, nervous function. I mean, even as much as touching the table, I've just sent a message up to the brain. So everything is neurofunction. So I'm going to talk today about changes in the dopamine system, but I'm going to pretend like it's everything I talk about today is very specific to dopamine, and it's not. We have some other changes in serotonin and things like that. So we know that the genes, or a lot of the genes that cause autism, actually impact neurotransmitter function and things like that. So anything else that can impact the same mechanisms can have that effect. And then, so there's an easy science like biological, and then there's also the input from other people, like just not getting behavioral feedback from parents and things like that. So there are a lot of ways, and if you can come up with an answer to that question, you are going to be so rich and famous, right? But in fact, there's probably 100 different answers, for sure. So. Okay, so where does all this come from? Why does this interest me, and where does the manganese come in? So anyone familiar with my research program knows that I really have a very deep-seated affection for vitamin C or ascorbic acid. It's an excellent antioxidant, but that's not just it. Because it's an antioxidant, it's actually a wonderful enzyme cofactor. So all it's doing is changing the oxidation status of iron for the most point. So Fe3+ and 2+ going between ferrous and ferric iron, and that typically changes a binding site on an enzyme, which makes it more or less effective, right? And so because of that, it's involved in neurotransmitter synthesis and a whole lot of other things. And so it has a lot of really important roles in the brain that people don't often think about, but I'm going to talk a little bit more about that. This is not a vitamin C talk today, I promise. But what's really interesting about manganese is a lot of the pathways are the same. So manganese is also an enzyme cofactor, and the two molecules really share a lot of pathways in common, which is probably why I took to manganese research so very quickly and enjoy it so much. But in my lab, we're interested in really a lot of different environmental factors that impact development of age-related degenerative disorders and also developmental disorders. So we have active projects right now relating to traumatic brain injury. We've looked at high-fat diets before and their effect on cognition. We look at sepsis and delirium, so bacterial infection and inflammation, and how all of these things can impact brain. So it does look like I can't make up my mind about what we want to study. But to me, a lot of these things are really the same, and they're chasing the same pathways. But today, we are going to focus on manganese, but I couldn't resist putting a little vitamin C in there, if only so that Professor Wilcox could roll her eyes at me when I pronounced it. All right, so I'm going to put the two up as comparisons, but also what I want to demonstrate is that very small changes in your environment, your diet, your exposures can have huge effects on brain function. So both of these are essential nutrients. Without them, you die. All right, and both of them come from dietary sources, and they're very easy to get enough of. So quite easy to be deficient in vitamin C. It's not as easy to be deficient in manganese because it really is It really is ubiquitous. But there are some SNPs, some single nucleotide polymorphisms, some mutations in the transporters associated with both manganese and vitamin C, that if you have these, it doesn't matter how much you take in, you can't use it properly. And so they share that. So it's easy for us to have very differing levels of both manganese and vitamin C in the body, in the brain. You can't really overdose on vitamin C because of the way it's transported in the intestines. If you take too much, it's just going to be excreted. Manganese is different, and that is the basis of everything that we're going to talk about today. So both of them are very important enzyme cofactors or co-substrates, and three of the main areas in which they work with a lot of different enzymes. So neurotransmitter synthesis, so we're going to talk about dopamine today. Oxidative stress and antioxidant activity, so vitamin C is an antioxidant. Manganese is very important in manganese-dependent superoxide dismutase, so it is a mitochondrial antioxidant. It catalyzes superoxide to hydrogen peroxide. They're both also involved in things like DNA methylation, so epigenetic changes, and then glycosylation is also fun. Glycosylation is just basically adding a sugar onto a protein, but that's going to massively change the function of the protein. So it could make your transporters work more or less efficiently. And so we think that's part of what's going on in the dopamine story. Okay, so here is dopamine synthesis, and simplified, I think. And I'm trying to demonstrate here there's a lot of places where manganese can have an effect. We have ERK at the top that needs to be phosphorylated. When it is phosphorylated, it allows tyrosine hydroxylase to be phosphorylated. That allows tyrosine to be converted to L-DOPA. Interestingly, ascorbic acid is a cofactor there. So you need ascorbic acid to make dopamine as well. When you've made your dopamine, so all those gray circles are actually enzymes. Each of those can actually be impacted, I think, by dopamine, particularly monoamine oxidase, which is the MAO there. And so the metabolism of dopamine, how it's broken down, there are several ways that ascorbic acid is involved, that manganese is involved, and then the transporter itself, so how you clear dopamine from the synapse once it's been released. We know that manganese can also affect the DAT, the dopamine transporter, either by glycosylation, moving it on and off the membrane, or some other function that we haven't quite worked out. So because of this, and this is all known, I didn't, work any of this out. Looking at ADHD, looking at dopamine-related neurotransmitter type developmental disorders is a really strong area for manganese research. So there is a lot of evidence, strong evidence, linking metal exposures to developmental disorders. We have clinical data, rodent data, primate studies. Manganese is not the only metal we're exposed to. We have ubiquitous exposure to environmental toxicants. There's at least 85,000 known. And look, the EPA tests for 300 of them. So I hope that makes you feel really safe and secure when you're out and about and drinking water. Right? Metals that we find in our water source, in our air, include lead, methyl mercury, so manganese, arsenic. People here were working on cadmium. Also iron. So some of these, iron and manganese, are required. Okay, they have essential functions in the body in low amounts. Others, like lead and cadmium, have no real function in the body. So when we take them in, they tend to use up the same transporters and impact our normal metal function. And those can be modify cellular processes and be stored, and we can't get rid of them, and they cause damage. So what's interesting about manganese, we obtain it primarily through diet. I said we don't usually become deficient. And we have what's called a hormetic or U-shaped, inverse U-shaped curve. So this is the Goldilocks phenomenon. Too little is bad and too much is bad. And there's this, I can use this. Okay, this optimal exposure range in the middle there is reasonably narrow. Over here, we're going to get manganism and neurodegeneration. Over here, though, when you just have had a little bit too much, what's going to happen? The idea is, probably nothing. No one's really worried about that, but that's the area that I'm worried about. Okay, so getting quickly so we can get to the data, prevalence of ADHD is pretty high, about 5%, but there's also probably misdiagnoses in another 5%. That could be people that just don't have access to being seen by psychologists to get a diagnosis, or they're better at masking their behaviors, or they just don't have access to medical care. And it typically involves hyperactivity, disorganized thinking, impulsive behaviors, and a lot of this is to do with dopaminergic connections between the frontal cortex and the striatum. So I just want to remind you, approximately 80% of enzymes responsible for glycosylation, they're dependent on manganese, and that the dopamine transporter is dependent on glycosylation for its function. So I'm not really going to focus on this much, but I just wanted to let you know there's a lot of ways that this dopamine transporter can be impacted. And we've got a lot of evidence. So we found higher manganese in serum and hair in children with ADHD. Areas that have high manganese, usually from pollution. So there's one study from an e-waste recycling center in China. Some form of behavioral deficit was reported in nearly half of all kindergartners in that area because there's so much pollution that they're exposed to. An Italian study showed that actually manganese transport impacted whether or not someone was going to have a diagnosis of ADHD, and that transporter mutation was the link was stronger in females. And then even here in North America, there was a Canadian study showing that just manganese in the water, not pollution, just normal water that we're drinking, the more there was in the water, the poorer the cognitive outcomes were in children. So this is not causing a developmental disorder, but this is maybe changing where you fall on the normal curve. And that's okay if everything's normal, but if you're facing some other challenge as well, it can be difficult. And this is a map of where manganese levels are higher in the States. So red is bad if you want to look at where you grew up. If you want to look at where you are now, are you in a red area or a blue area? Places like Minnesota, which I like as MN, just like manganese is MN, actually have pretty high manganese in their water. And so their government has issued, local governments have issued warnings about making formula. So formula-fed babies are probably okay if they're on a milk-based formula, but you can see a soy-based formula might have 5 times the amount of manganese in it. And so when you make that with a manganese-heavy water, you can actually be overdosing children pretty easily. So all of this to say, it's probably not going to kill you, don't worry, but it is possible to get higher than optimal amounts of manganese and other metals in your water pretty easily. Now, the question is, does it matter? So some of the questions we're asking is, does manganese exposure cause behavioral changes associated with developmental disorders? And a lot of questions come from that. Are some groups more affected than others? Are boys or girls more affected? Does your genetic background matter, what you're at risk for anyway? What is the mechanism? Can we fix the damage? How do we impact that damage? Can we prevent it? And then really we're asking, is that dependent on DAT function, the dopamine transporter? So how are we doing this? So Adriana here is my lab manager. She runs a lot of the studies we do in the lab. And then Ashley here is a current high schooler who's helping code a lot of these videos. So we used this mouse here, this DAT T356M mouse. So this is a mutation that was discovered in an individual with an autism diagnosis who also had repetitive behaviors and hyperactivity. And this mouse model was generated by collaborators of ours, and we're using that for this study. And what we're going to do is we're going to put these mice on a long-term diet, either normal levels of manganese, so 70 parts per million, or a very high level of manganese, 2,400 parts per million. Not toxic, it's not going to be killing off cells anytime soon, but just much more than they should have. All right, so this is the dopamine transporter mouse. And what you can see here is severe hyperactivity. So what happens is dopamine is released into the synapse, and it should be cleared quickly by the dopamine transporter. But this mutation is just a single point mutation, a threonine to methionine change. That's where the position 256, that's where the name comes from. And it causes the transporter not to transport as well. It actually becomes leaky. It can kind of reverse direction a little bit. So less is cleared and some leaks out again. That means the dopamine is active in the synapse for longer, and these mice are very hyperactive. So this is quantification of mice just running around in an open box. And you can see the red, the homozygous mice are really hyperactive. So that's them up here. They just keep running. They're just going like this. If you put them in the home cage, they're going like this and they're climbing. They also have a lot of these behaviors, these climbing and rearing and grooming behaviors, and I'll talk a bit more about those later. Now, these are the same mice, and it turns out if you use specific inhibitors on the dopamine transporter, you can decrease the activity. So we're pretty sure that a lot of the change here is really because of this mutation in the dopamine transporter. So what we did was we took heterozygous mice, so not the homozygous mutant. We used just a single mutation and one normal copy. That's the case that humans have, right? We're not usually homozygous for the gene mutations we have. So we were expecting a much more subtle phenotype. They should be just a little bit hyperactive, but that allows us to see changes based on environment on top of that. So the first thing we did was we looked at locomotor activity. So we just put them in a box and let them run around. And we did this at baseline, then we put them on the diets, and we tested them every 4 weeks. We looked at spontaneous behaviors. We put them in a jar and we just watched them on video. And Ashley and some other people in the lab have just stared at videos of mice and coded them for all of these behaviors. It is a painstaking, I won't say thankless task because I do thank them, but it is a very long, drawn out task. We did some motor learning and then we did a drug challenge at the end. I'm going to point out one thing here. We have some rules which we're supposed to observe about not posting photographs of mice. So all of the mice I have are actually AI generated by DALL-E. So if you see anything really weird on these pictures, that is why they are not real. I will point out some of the weirdities as we go through. All right, so this is what we do. We put the mouse in a box. So again, it's AI, so this box is broken and it's a very small box. In fact, the box we use is about this big, it's about 30 centimeters square, and we just let them run around, all right, and do what they want to do. They usually spend more time exploring the edge in the center where they feel a bit safer, but they'll explore the whole thing. And we look at their path length and take readings from that. And so as you can see, the heterozygous mice in gray are a little bit more hyperactive. Not all of them, right? But as a group, they're a little bit more hyperactive. So this is a really important behavior for mice. Their entire existence depends on exploring their environment, getting food, finding a mate, and being happy, but also knowing when threats are going to be in place. So exploration is a very key behavior for mice. It's also dependent on dopamine signaling. So after they've been on the diets for 4 weeks, you can see that the heterozygous mice, which are now over here in the gray, are still hyperactive. So this is the males. So this bar here is showing us that those are significantly increased in behavior. Now in the females, the heterozygous mice had increased behavior, but the females on manganese had a significant decrease. So something about that manganese diet is changing activity in the mice. So we actually had predicted the opposite. So I was kind of surprised to see that. After 8 weeks, we looked again, and now manganese in the males is increasing activity. Manganese in the females is decreasing activity. And we thought, well, that's odd. Behavioral data are always slightly variable, but I wasn't expecting to see such a large difference. And then by the time we got to 12 weeks, they've been in these boxes a lot. They're not very interesting to them anymore. We saw less changes according to manganese. So basically, we have manganese increases activity in male mice, decreases activity in female mice, but the effect does diminish with either age of mice or time of exposure. I don't know what was causing that change. Can anyone tell me what's wrong with the grooming mouse at the top? See it? I can't hear you all yet, but yeah, he's got an extra paw. Yeah, DALL-E is hysterical if you try and make photos. It's really good. But yeah, so this is what we're coding. So we're looking for when they're doing grooming behaviors like this or just scratching. So apart from the extra hand, these are pretty good pictures. And what we found was after 4 weeks on diet, grooming was decreased in male mice on the diet, but not affected in females. And remember, this is the sort of behavior that we see in models of developmental disorder. It's a repetitive behavior, but it's a very mouse-centric development, repetitive behavior. It's also can be an anxiety behavior, a sort of almost like a comfort thing for mice. By 12 weeks, there's really no change. So again, manganese is doing something in the males, it's decreasing grooming, and there's no change in the female mice. So we thought, okay, well, maybe this is right, that we're getting different results in both sexes. So then we looked at rearing and climbing. Okay, so the prompt here, we put them in a jar, in like a glass jar like this. So I asked it to do a mouse standing on its hind legs leaning against a jar, which technically it has done here, but that is clearly not what I meant. But this is what we're looking for. The mouse will stand up and do this. So it's exploring, it's looking at its environment, it's interacting with the environment around it. And what we found was that at 4 weeks of diet, male mice, the DAT mice, do this more, and we had observed that in the homozygous mice as well. But manganese really potentiates that effect. In the females, we have the genotype effect. We don't have an effect of manganese. That genotype effect stays in the male mice. It diminishes over time in the female mice, but that effect of manganese wears off. So, I mean, I thought that's really interesting. And I thought, oh, well, all of our effects are wearing off that's maybe the age of the mice or the exposure. Maybe something is adjusting in the mice to deal with this high exposure at some kind of homeostasis. So everything we do now, we're not going to see any differences. Turns out I was wrong. I'm usually wrong with my hypotheses. That's okay. That's why we get data. So this is nest building. This is the only real picture. What we do is we give the mice these nestlets and they can shred them. So nesting is a very important behavior for mice. They need it to survive, have their young, keep themselves safe. And so shredding is also a repetitive behavior, or it can be. And so they should build lovely nests. They should shred this overnight, build themselves a nest. But sometimes they do, and here in this case, obviously they don't. We think that to make these nests, because we can look at lesioning studies and change function in the hippocampus or different areas of the cortex, we think that this actually involves a mouse version of executive planning to do this. It's also just a naturalistic mouse behavior, repetitive motion for shredding, something like that, motivation. So even though it's a really cute little task, there's a lot of sort of cognition potential in here to explore. So a mouse that moves this nestlet around, maybe takes it over to the corner, might score a 1. And a mouse that forms a beautiful igloo where you can't even see the mouse is going to get a 5. Males were not affected at all. Females, this is the score over here, manganese actually made them better at building nests. And we thought, well, that's interesting. Why is that? And if you weigh how much they shred of the nestlet, so how much is left as a chunk, because sometimes they'll leave little chunks, but they'll use them structurally in the wall. It's actually very clever. But the female mice on manganese shredded everything. You just couldn't stop them. So we're thinking maybe this isn't better executive planning. This might be more of a repetitive, compulsive type behavior in the mice. But again, we have sex differences in how manganese impacts these animals. And like I said, I thought there wasn't going to be much difference now because the manganese effect seemed to have worn off. But it was really interesting that we had males and females behaving differently, even though some of these changes are kind of subtle. So I thought, well, we'll try an amphetamine challenge. Why not? We know that the homozygous animal is actually resistant to cocaine. So the dopamine transporter DAT is actually a target for cocaine as well. So it can act as a competitive inhibitor. So it stops clearance of dopamine because the cocaine is there at the transporter instead. And that's why you get dopamine longer at the synapse and you get hyperactivity in the mice. Now, because the mutation in the DAT mouse is closer to the binding site and alters the binding site, that competitive inhibition doesn't work as well. It doesn't happen. So cocaine didn't have the same effect. So we thought, well, we'll try that with amphetamine. So what happens, because amphetamine will bind the DAT, and I'll talk about that a little bit more in a second. So we put these mice, they've been in these chambers a lot before, so they're very habituated to them. So they go in for about half an hour, just to make sure that they're not exploring a whole bunch. Everyone's now pretty much exploring the same amount. Then we give them amphetamine here, and you can see the effect. They just start running. They love it. Whee, around we go. They're kind of harder to pick up at the end when you try and catch them. And then what we've got here is a quantification, an area under the curve. So what was that effect of amphetamine? And what you can see is in male mice, manganese has potentiated the effect of amphetamine. So that line there is a little bit higher up, kind of in both groups a little bit. But you also see these are the heterozygous mice down here. They just don't really respond to amphetamine the same way. So that was what we were expecting to see. But this effect of manganese was interesting to us. Now, in the females, what's interesting is even the heterozygous females are kind of a little bit responsive to amphetamine. So there's something about the way this mutation is expressing in males versus females that really starts to lend some credence to the funny findings we've had for the rest of this experiment. But manganese kills that response. There is absolutely nothing happening in the manganese-fed mice. And remember, this is not a toxic, they're not sick, but it is definitely changing their response to drugs. So again, manganese potentiated the effect of amphetamine in the male mice. Manganese decreased the effect of amphetamine in the female DAT mice. And just to go through the biochemistry of this a little bit, they're both stimulant drugs, okay, amphetamine, methylphenidate, so names that you recognize are Adderall or Ritalin. They're used to treat ADHD and increase attention and focus. And there are a number of different mechanisms through which they do this. Some of them are the same, some of them are different. So they bind to the DAT. They can be taken up by the DAT as well. So that disrupts clearance, leads to increased dopamine at the synapse. So that can help with attention and behavioral differences if there's a deficit there in dopamine. And obviously, if there's too much, it's going to increase activity. So that's why these have different effects in people with ADHD and people without. So they also amphetamine disrupts storage in the presynaptic vesicles. So more dopamine is available in the neuron. You can actually cause the dopamine transporter to go the other way and release dopamine. So a bit like our mutation causes it to be leaky and release dopamine. And methylphenidate does many of the same things. There's also, both of them can kind of stimulate some other receptor types as well. So there are other neurotransmitters playing in here, but the main effect is dopamine. And so just to remind you, the dopamine transporter mutation we're looking at impairs drug binding at the DAT. So it's going to limit uptake, limit response to the drugs. And then manganese, so what is that doing? I think it's involved in dopamine synthesis. We looked at that diagram earlier. We think it's involved in dopamine metabolism. So those are both through changes in enzymatic function. We think it's involved in the function of the dopamine transporter. Maybe its location on the membrane. That's going to be glycosylation changes, again, enzyme functions. And then it actually also might be involved in dopamine oxidation. So this is a metal that's around. There's going to be more oxidation. There's a lot of oxidation involved in Parkinson's disease, things like that, where dopamine is damaged. And dopamine is very easily oxidizable. So that could be some way it's coming in. But we thought, this was a big effect. And I thought it can't be that good, right? So we'll do it again. All right. So we changed the experiment a little bit, just to be sure. Krista Paffenroth, who's our behavioral technician, did this. So we have the same mice, but now we put them on manganese supplements at the age of 3 weeks, just when they start to be weaned. And then they stay on them until 7 or 8 weeks when we do this study. And now we have the same kind of idea, normal or high, but not toxically high. For this, manganese makes the water taste metallic. That's, by the way, why we have guidelines in place for human manganese in water. It's because it tastes kind of rusty and metallic, not because it's dangerous to you. So we have to put stevia, we have to put a sweetener in the water or the mice won't drink it. We have to put it in the control water as well to make sure that everything's matched. The control mice love this. They drink all day because they love the sweetened water. And so the design here, they get 3 saline injections before going into these activity chambers. And that is to cause habituation, because I want to minimize the effect of that hyperactivity due to the mutation. And I want them to get used to being jabbed in the belly, right? So that any result I see is an effect of a drug and not an effect of, oh my gosh, somebody just stabbed me with an injection. Okay. So they get 3 of these. Then they get either amphetamine or methylphenidate. Yes. Oh, that's a really interesting question. Could having the sweetened water being a pleasant thing lead to changes in dopamine is the question. I mean, actually, maybe it could. I've never looked at that. And the reason we use water is because we're talking about exposure to people through water, so it does make more sense to do it through the water. I don't think there's any difference in terms of uptake, whether you put it in diet or water, and diet is a lot easier. But really, that was a response to reviewers and the the community who uses this kind of research tool suggested water, so that's why we do it. And we use stevia, so it's a non-calorie sweetener and things like that. I worry a little bit about sweeteners in the water, but it is in both groups, and we have not looked at whether the stevia itself changes that. That's a really interesting question. Okay, so we have our 3 habituation sessions. We have amphetamine or methylphenidate. Then we have a week washout. It probably only needs 24, 48 hours to completely wash out, but just to be sure, we give them a week. Then we do another baseline session with saline just to make sure everything's back on track, and then we do the second drug. So this is the baseline. We can see that we have in the males hyperactivity. We saw that before. We have that over the second, and then by the third session, it's about normal. In the females, same thing. They're still a little bit hyperactive. But again, manganese decreased activity in females. So I like when I see the same result despite slight changes in the experimental setup. So that's experimental rigor, right? Doing it different ways and getting the same result. And that really makes you feel better about what you're finding. So then we gave them the drug, and oh my goodness, we found essentially the same results again. So in the male mice, you can see the wild types here in the black with the white, and then the potentiation. So here they get the, at time 0 is where they get the drug. That's why it goes straight up here. So a huge potentiation of the effect of amphetamine, and then no effect in the DAT mice. And then that's quantified down here using, I think, area under the curve. And then here in the females, we see that the DAT mice are partially reactive, but not totally reactive to amphetamine, and there's really no effect of manganese. So that was the same result as we saw previously, that manganese potentiates the effect of amphetamine in the males, particularly the wild-type mice, no effect of amphetamine, of manganese in the females. We moved on to using the methylphenidate. And what you can see here, it's a little harder to make out the groups, but this is the wild type down here. And you can see that manganese causes a potentiation of the effect of it's not amphetamine, actually, it's methylphenidate at this point. In both the mutant mice, the filled in, as well as the wild types, no effect in the females. Okay, so again, we've got these drugs are being altered in male animals, but not in female animals. And I'm just keeping an eye on the time, but you may recognize this dewy-eyed graduate student here in the corner, now known as Professor Wilcox. So we luckily already have a little bit of data on this. And what she and this other student, David Consoli, had done was look at injections of manganese over 3 days. And that's a way that we can very quickly increase manganese in the system. And so that treatment schedule decreases dopamine and decreases the metabolites. But more importantly, we wanted to learn a bit more about the kinetics of the dopamine transporter. So this is fast scan cyclic voltammetry. So dopamine is easily oxidizable, and you all remember your chemistry here. So oxidation reduction is just gain or loss of an electron, and that changes the current. So as you oxidize dopamine, you change a current, and you can actually use this change in current to determine how much is present. So we take slices, live brain slice. Well, the mouse is dead, but if you do this very quickly, the slice is still alive. We keep it going in artificial CSF. And then using a stimulating and recording electrode in the nucleus accumbens, which is down here, which is a very important center for dopaminergic transport and function. You can see you get when you stimulate, you get a release of dopamine that gives you this peak here. And then as that is cleared, this peak changes. So we can get a measure of release and a measure of clearance. And it turns out that manganese does not affect release of dopamine necessarily, but it really does affect clearance. So these purple mice are actually a model of Huntington's disease that does not transport manganese appropriately. And so what's really interesting about this is you can see the result. These mice aren't involved in this study, but you can see that they don't transport manganese appropriately, and the effects are different. So they're in fact reversed. So we know that manganese can impact dopamine transport. Dopamine, it does not affect dopamine release. It does decrease uptake. So again, further implicating the dopamine transporter. So what is the potential translational value of this? Because I think mouse behavior is super exciting, but apparently people think children and adults are more important. Fine. So what can we do about it? Well, I would love to say, look, let's limit manganese exposure in at-risk populations. Let's write a paper, let's publish these results, and let's show people that they should clean up the water sources. But it's probably not going to happen anytime soon, right? But maybe we can use this knowledge in some way to perform individualized medicine approaches. So these sex differences are just fascinating to me, in particular. So can we measure manganese in everyone? I mean, technically you could. It's kind of tricky to do. You have to get everything on the mass spec, but it's fine. You can do that. There may be some other reliable biomarkers for manganese exposure. So we can look in blood or tissue. You can get long-term exposure from hair or toenails, which is disgusting. But I spoke to a graduate student who was running that study a couple of weeks ago. We can also look at manganese responsive genes, probably not one or single gene, but a combination of genes of all these enzymes that are actually responsive to manganese in the environment. And we can get an idea of what somebody's lifetime exposure was. We can also use imaging techniques. So memory or manganese enhanced magnetic resonance imaging works because you can see manganese on an MRI. So it has paramagnetic properties that change. So MRI, you're using magnetic fields to align and relax water molecules. I'm not a physicist to do this, but because manganese has these paramagnetic properties, it changes how the molecules align and the speed at which they relax, and so you can see it. So you actually can use this by memory is a technique where you inject manganese at high concentrations, and because manganese then substitutes for calcium, it's going to show up in cells that were recently active. But you don't. That's a research technique. You wouldn't go and inject patients with a whole lot of manganese. That would be really bad for them. But it turns out that if you use patient samples, because of this property of manganese, you can see it. So this is an example of high exposure in the welding industry. And so these white areas here, this is manganese deposition in the striatum, in the caudate putamen there. And so manganism looks a bit like Parkinsonism, gait and movement changes, rigidity. There are, you've got here slurring of speech. There's also personality changes that go along with that. And you have this manganese accumulation. So we wondered, well, we can see a lot of manganese. Can we see a little bit of manganese? And so we started to look at this in the mice. And so I'm just going to give you a taster of where this research is going, how I hope to use some of these data. So you can, I think, appreciate here that the brains on top are yellowish and the brains below are bluish. All right. And so the yellow-blue is the T1 relaxation time, and the presence of manganese makes that relaxation time shorter. So the bluer the brain slice, the more manganese is present. And so these are brains from that last pharmacology study. And it turns out that the brains on top are in the control group and the brains on the bottom are in the high manganese group. So to the eye, I can see differences and I can pseudo quantify what's in there, or at least we're trying to. And so in fact, the one on the bottom right is, I think, the heterozygous animal. So there may even be differences in, the wild type and the hets, but we haven't got that far. So we're actually quantifying from the black and white images. I personally can't see differences in the black and white images, but that's what we use for quantification, and that's how we identify areas of the brain to look at. So how we quantify it, so up here or on the bottom, the top one is a control, then we have a diet tested, and on the right is an injected, which has the highest, and again, you can see this. And so when we quantify from multiple slices. So we digitally slice the brain in 100 micron sections. And then we can quantify completely different areas or nuclei of the brain, so we can get a reading throughout. And right now, we're painstakingly have a graduate student going through hand coding different areas, but the idea is to use an AI-based image to take our slices, bring a 3D reconstruction back to the brain and quantify manganese throughout. So that will give us an idea of exactly where it's accumulating, exactly what cell types, and what processes it might be affecting. And then what can we do with that data? I wanted to take it one step further. And so we've tried to use clarity. So brain clear, it's actually not clarity, it's a different version, but brain clearing, what you do is you dissolve the lipids out of a substance. And now they've got to the point where they can actually do this in a whole mouse brain. And it allows you to image all the way through the brain. So we're not restricted to just slices. So this is kind of hard to make out, but this is because I'm taking it on a corner, you can see it from two different angles. It's the same thing from two angles. So this is the brain right here, and you can kind of see through it, but you can't even see it here because it's actually clear-ish, right? And then what they do is this is a horizontal digital slice through the brain. And that's taken at very, very thin slices all the way down. So this is done in our neurovisualization core, and we're just starting to get the data back, again, from these same mice that we did the pharmacology study on. So I'm going to show you a video if it plays, I hope. But what you're going to look at is this. So the yellow is tyrosine hydroxylase. So this is going to mark dopaminergic neurons. And what we've also done, which is not in this video, is use CFOS, which is an immediate early gene, showing cells that were active. So we will be able to say in which area of the brain, depending on how much manganese there was, were cells more or less responsive to manganese, and were those cells dopaminergic cells. And so right now this is just, a research cool technique, but I think it's going to give us a lot of information. Can we see that? All right. So we're starting, this is the center of the brain. You can see the cell bodies, you can see projections. This bit on the left is actually probably a blood vessel. You can ignore that. But you can see these cells, and we're going to zoom out now in a second. So those lines are the projections coming from each of the neurons. And I think you can appreciate that there are areas that are very, densely populated with dopaminergic neurons, and there are areas that are less populated. So the front of the brain here, we're back to the olfactory bulbs at the bottom right. The white is background, but actually that could be helpful sometimes to orient us. So that is a digitally reconstructed 3D brain. So the goal is to put these images together. Now, I don't know how to do this programming. But with the new AI approaches and data approaches that are coming online now, probably any undergraduate can walk into my lab and be like, duh. Done. And I think that there's a huge amount of power, because what I can then do is take these approaches into the electronic medical records, where we have a lot of MRIs. We have people's whole health histories. We have biomarker information. We have genetic information. And so we actually could start getting at individualized approaches to, what, you live here, you grew up here, I'm going to start you on a lower dose, and you're not going to get the same side effects. For you, this drug is not going to be effective. As a female, you're going to need this instead. And so using this information, I really think we can start to improve how we're treating people. And I like to freak people out at the ends of these talks by saying, no, don't worry, you're fine. I looked up the water right here in South Bend. And don't worry, because they only found that 10 things in your water exceed the EWG health guidelines. And of all the things that don't exceed it, you don't need to worry about that. Well, actually you do, because it turns out that just because the government says it's not going to kill you doesn't mean it's optimal. Someone asked me at lunch, like, what has my research changed? I don't filter my water. I mean, I don't think you can escape everything, but this is also always just kind of fun, right? So what's in your water? Arsenic, all right, above health levels. Nitrate, hexavalent chromium, that's from the Erin Brockovich story, right? Probably not at the levels that they had it. Other contaminants detected. Almost always I find manganese in there. So you're within safe levels, okay, for manganese, but it is definitely in your water. All right. And just I want to point out, federal government, there is no legal limit for manganese, by the way. There are some non-enforceable guidelines, mostly based around that rusty taste that you might get. And for a lot of things, I think it's just worth being aware that the guidelines aren't always as strict as we think they should be. All right, so in conclusion, chronic dietary manganese is sufficient on its own to alter behavior at subtoxic levels, right? Not cell death, anything like that. Manganese-induced changes, we think, are via dysregulation of the dopamine transporter in some way. But also, brain manganese might actually determine response to commonly used drugs, for developmental disorders and who knows what else. We're testing some Alzheimer drugs as well right now. Importantly, these exposures have different effects on males and females, may have different impacts according to age. We actually get different results depending on the type of mice we're using, so your whole genetic background has an effect. And then these were at levels that were not acutely toxic. So just finish with thanking absolutely everyone in my lab. I put an older picture so that I could put Professor Wilcox on here. I thought she was going to say meaner things about me during the introduction. So, but yeah, it's my lab that does all the work, and Dr. Erin Calipari is my drug person. So she has been incredibly helpful setting these up, and we use a lot of core facilities at Vanderbilt. So I'm very close to my time. So thank you, and I will take questions happily.
Have you thought about doing earlier exposures during early mouse development and/or giving these manganese doses to the female mothers while they're pregnant?
Yes. And it's a fascinating story, and it's a whole different story. So we had a postdoc with us for a while, and then she left, and she had started getting some data on this. And we exposed the mice, again, to these non-toxic levels during pregnancy. And the mice, and there's a lot of data about changes already. I would love to do that and look at the drug response later on. But these mice were wonderful because they were born normally. They developed normally about until postnatal day 10 or 11. All their fur grew in, they're cute, they're running around. And then they started to lose their fur, and they lost it from the back out. And so by the time we weaned them, they had fur on their heads, and they had a little butt diamond. They had a little diamond above their tail, and the rest of them was bald. I don't know why. But we only did a couple of litters that came out that way. And so I have no, and I'm sure if we've got skin effects, I'm sure there are going to be neural effects. But they were healthy, just bald. So yeah, there's bound to be huge effects with prenatal exposure.
Yes, interesting presentation. I'd like to know what kind of vitamin C did your colleagues use in the study? Did they use regular, like nature-made, God-made vitamin C from lemons and limes, or did they use man-made ascorbic acid that was made in a lab? It's usually made with genetically modified corn. Do they stipulate which type of vitamin C? I mean, both could be called vitamin C, but the man-made type can be just legally called vitamin C. So did they stipulate which type of vitamin C they used in their study?
It actually doesn't make a difference to my mind. And so typically we're using synthesized ascorbic acid, and the body's going to go ahead and convert it into ascorbate, and we're all good. So the benefit of having nature-made vitamin C from a fruit or veg is that everything else is in there as well, right? So there are clearly benefits. But when we're studying one thing at a time, we typically use a much more controllable source. And to my mind, vitamin C is one of those things that it doesn't matter where you get it. We do have some slightly different forms that we use when we're doing cell culture studies, because it'll actually keep active for longer. There are sort of different phosphate groups that you can stick on. So there is some chemistry. And we're doing some really fun studies with this fruit that I'm not supposed to talk about from Brazil, that has very, very high vitamin C, but a whole bunch of it. And the person who's having me do this believes it's the vitamin C, and I don't think it is. I think it's the vitamin C with everything else as well. And so I am if you're hinting that we should all be getting this from food, I agree with you. Like, that's clearly the healthier option, but chemically speaking, it doesn't make a difference to my mind at all for that. Some things it does, but ascorbate it doesn't.
First of all, thank you very much for the talk. Interesting.
Thank you.
I had a question about just the social behavior. Did you guys do any experiments on that, like in regard with the males and the females, or males and males in general?
So we did social behavior in the homozygous knock-in mouse in that original study. And it was difficult though, because those mice were just crazy. So it's really hard to do when the mice have so much hyperactivity. But we look at their preference for hanging out in an area where there's another mouse versus an area that's empty. We also looked at social dominance, and this is a fun task. It's called the tube test. So you put a mouse in either side of a tube. They go in, they face off in the middle, and essentially whoever's the dominant mouse just kind of says, back off, and the other guy goes, oh, okay. And it takes 10 seconds to 2 minutes. They don't fight, they don't even touch, but it's a very reproducible behavior. So that was impacted in the dopamine transporter mice. It's tricky to do social tasks really well. There's a lot of nuances. So we didn't do it in this study. We focused on the behaviors that we were pretty sure. We did a couple other things I didn't show, but we focused on the behaviors that we were pretty sure we were going to see clearer effects in. But it would not surprise me at all to find that we still had differences, and that might be affected by the manganese as well. Anyone else?
So we can all give Dr. Harrison a wonderful round of applause.
Thank you.